Hey everybody, I wanted to talk about the Krebs cycle, also called the citric acid cycle, with you. First, let's look at a quick overview of cellular respiration to see where the Krebs cycle fits in here. So the first step in our process is glycolysis, where you cleave a molecule of glucose and you end up with 2-pyruvate or 2-pyruvic acid, same thing. You also end up with some reduced coenzymes and 2-net ATP, but these 2-pyruvate are what will head into ultimately making acetyl-CoA and then into the citric acid cycle. From the citric acid cycle, all of these reduced coenzymes that you've made will enter the electron transport chain. So we're kind of in this middle section area talking about the Krebs cycle. Just a refresher on mitochondrial anatomy. Remember, glycolysis takes place in the cytosol, but those pyruvic acid then will enter the matrix of the mitochondria, and this is where you'll form acetyl-CoA, and it is also where all of the reactions of the Krebs cycle will take place. It takes place in the matrix. So if you remember, mitochondria have an inner and outer mitochondrial membrane. The inner membrane has all of these folds in it, and so the matrix is going to be the innermost space right here, innermost space. So this is where formation of acetyl-CoA and the Krebs cycle will take place. Pyruvic acid, which we got from glycolysis, is a 3-carbon molecule. Each carbon is symbolized by these gray dots right here. The first thing you're going to do is take off a carbon, and this carbon will be removed and bond with oxygen to form carbon dioxide. So here is carbon dioxide, metabolic waste product. You have 2 carbons left from pyruvic acid after taking that one off. Now you're going to oxidize it. This molecule will be oxidized, which means it will lose electrons. Remember, with redox reactions, oxidation is losing electrons. Reduction is gaining electrons, if you remember your oil rig. So this 2-carbon molecule will be oxidized. It loses its electrons to become an acetyl group. NAD will be reduced. It will receive those electrons to become NADH plus H. So this 2-carbon molecule was oxidized and NADH is reduced. So the electrons moved from here to here. This 2-carbon molecule is oxidized. NADH plus H is the reduced form. So this acetyl group is now going to bond to coenzyme A. Coenzyme A, when it receives this acetyl group, these 2 carbons, becomes acetyl coenzyme A, or acetyl-CoA for short. Acetyl-CoA is then going to donate these 2 carbons into the citric acid cycle. But this coenzyme A is not going to be used up. So here's where we came from: acetyl group, coenzyme A, acetyl-CoA, donate those 2 carbons into the citric acid cycle. I'm going to move over here to our bigger picture. So here is oxaloacetic acid. You can see it is a 4-carbon molecule. When you add in these 2 carbons from acetyl-CoA, it becomes citric acid. So this is why the Krebs cycle is also called the citric acid cycle. Citric acid is a 6-carbon molecule, and you do not need to worry about the names of these molecules or the names of the enzymes that are responsible for converting these molecules for my class, with the exception of oxaloacetic acid and citric acid. You should know those terms. Otherwise, just know that the Krebs cycle takes place in the mitochondrial matrix. It does require oxygen, so these reactions cannot take place when there's an absence of oxygen in anaerobic conditions. You must have aerobic conditions. But otherwise, this molecule will continue to be rearranged. Some carbons will be removed as carbon dioxide, and then as you work your way back around, you have these carbons added back on thanks to acetyl-CoA donating those carbons. But the most important thing that I want you to know about the citric acid cycle is these reduced coenzymes that are made. And you see this several times over the course of these reactions. So as this molecule becomes modified, release a little water, here's a reduced coenzyme right here. So you're going to remove electrons from this 6-carbon molecule. They are going to attach to NAD to become NADH plus H. So here is your reduced coenzyme. This is one of the most important products that you get from the citric acid cycle. Remove another carbon. Now you only have 5 carbons. You're going to reduce another coenzyme. You see the same thing occur. Take off another carbon, and over here you see you're going to generate one measly molecule of ATP, and this is just through substrate level phosphorylation. Continue to modify these carbon-based molecules. FAD becomes FADH2. Here's another redox reaction. Oxidize this molecule, reduce this coenzyme. Water comes into here, another reduced coenzyme right here, and you're back where you started, oxaloacetic acid. And now you're ready to receive 2 more carbons to become citric acid, and you can go through this series of reactions again. So you keep, we refer to it as turns of the citric acid cycle. With every turn of this cycle, for every individual acetyl-CoA that donates its carbons, or every individual pyruvic acid, you make 1, 2, 3 NADH plus H, 1 FADH2, and 1 molecule of ATP. This is just for one turn of the citric acid cycle, or the Krebs cycle. Remember that for each molecule of glucose that goes into glycolysis, you make 2 pyruvic acid. Going back to this picture on the left side. 2 pyruvic acid will make 2 acetyl-CoA. So here's 1 acetyl-CoA that's carbon going through, and then the second one will head into the Krebs cycle. So if you're asked for each molecule of glucose, how many of these products are produced, then you'd say 2, 4, 6 NADH plus H, 2 FADH2, and 2 molecules of ATP. So that's how much is produced per molecule of glucose. Remember, it is split to make 2 pyruvic acid. So you have 2 turns of the citric acid cycle per molecule of glucose. I hope this